This conference will now be recorded.
Divya, I cannot hear you. Hello?
Now is it okay?
Yes, now I can hear.
Yeah, ma'am. Now it's okay.
This conference will now be recorded.
Yeah, good evening. I'm Divya from TeenEinstein. So today we are moving to next unit, not next chapter, next unit. So in the last unit, you had learned about reproduction, and then we had gone through different chapters with the same concept, right? So we have learned about reproduction in organism, then sexual reproduction in flowering plants, then human reproduction, and last chapter was about reproductive health. So these many things we learned under last unit, which was reproduction. Okay, so that's about the first unit with 4 chapters in 12th biology. Second unit, that is it is genetics and evolution. Okay, so here what is genetics? It is the branch of biology. So in this branch you can learn about genes, then genetic variations, then heredity in organisms, how one character is transferred from the first generation to the second and third generations. All this you can learn in this chapter or in this unit. So in 19th century onwards, all these studies are going on. Okay, so the branch of biology which deals with the genetic inheritance and variation, that is genetics. Okay, so that is the main pillar of biology, one main pillar of biology. Okay, so genetics, that is the base, one among the base of biology. So because of yeah, can you hear me? Why genetic areas of biology? Because it is merged with biochemistry, biotechnology, then agriculture. All these fields, all these are related to genetics. Our genetics is merged with all this subject. It is we can't avoid, we can't tell genetics without the help of biotechnology or without the help of finally chemistry subject, because all these they are coming to the same concept. So we told about genetics and evolution, right? In this unit we are going to discuss about genetics and evolution. So now what is evolution? Evolution is the changes in the heritable characters. Heritable means which is passing from one generation to another generation. So what and all the changes that in the heritable characters of a biological population, and how these changes will come over so many generations, or it will take so many years, it will take time to develop these changes. So these changes in the heritable characters of biological population, that is called as evolution. So for example, we can tell evolution of human, then hope you have learned in the 8th or 9th standard about the evolution in birds wings pattern and all. So how it formed, that structure, that structural changes, what and all came. So that evolution, that human evolution, everything you have learned. So under this genetics and evolution unit, we'll learn mainly 3 chapters, which explains about this concepts of inheritance, variation, then changes, or how it is evolving. So all these topics are there. So under this, we are going to learn about principles of inheritance and variations. That means what are all the principles or the rules that regulating this inheritance and variation, what are all changes, or how the changes are coming, or how the characters are going from one generation to next generation. Then molecular basis of inheritance, what are the molecular reason for that inheritance, that also we are going to learn. Then we are going to learn about evolution. So now we are moving to our first chapter. First chapter of this unit, chapter 5, principles of inheritance and variation. Okay, so principle inheritance and variation, in this slide itself you can see here the picture of plants. Here you can see the parental one, P1 and P2. We have marked as P1, parent 1 and parent 2. Parent 1 is tall and parent 2 is short. In the first generation, you can see the next generation produced. In the next slide, the next generation F1, which I have mentioned as self grows. It is self, these terms and all you can learn one by one. So in the first generation, you can see tall plants. So how one tall parent and short parent, these generation from parental generation, how all next generation plants are becoming tall. How that character is coming from parents to next generation, or parents to offsprings. Here one is tall and one is short. How it became tall in the first generation. And when it is coming to the second generation, here I have mentioned as F2. In the second generation, here you can see out of 4, just we are considering only 4 plants here. In that, 3 are tall and 1 is short. And what is the reason behind this? How come 3 became tall and 1 became short? In the same case, in animals case also. Here you can see the dogs and the puppies. See, here these are the offsprings, characters, and learn. In both cases, there is a mixing up of all these characters. We are from the parents, some characters. You can see some characters are inherited to the next generation. Next generation of this offspring, or the young ones are getting copy of the parental generation. Some characters are mixed up. So in this chapter, we are going to know the principles of that inheritance, or the roles or role of each character, or the genes behind it. We can learn about genes, then the genes which are the main factor for this characters, or how from parent one character is going to next generation. What is the reason behind it, and what are all the rules behind, or how it is getting shared to the next generation. So all this we are going to learn. So we are going to learn how these characters are coming to their offspring, and why there are some changes in parents and offsprings. Here you can see so many changes in the parents and offsprings. So why all the characters are not getting copied from parents to next, or what is the reason for that change? So that is what we are going to discuss in this chapter. Okay, so before going to the details, let's discuss about some important scientists who made major contributions in their genetics field. Okay, so these are the important questions for NEET. You will get so many questions from this part. Just note down all these points. These are important MCQs about the scientist and their major contribution in the field of genetics. Okay, the name of scientist and their contribution, their important contribution that you have to note down. So the scientists who made major contribution in this, first one is John Gregor Mendel. John Gregor Mendel. Genetics. With John Gregor Mendel, because he made a base for the subject. Okay, he made a base for the genetics. From his all this genetic studies starts. Okay, so he is the father of genetics. And all other scientists, they followed his path and they gave a complete idea of this genetics. Okay, the subject. In molecular level, they conducted studies in biology. In the level of molecular physics, they conducted studies. So all these came after the contribution of Mendel, Gregor Mendel. Okay, so the foundation stone was laid by John Gregor Mendel. Okay, so Gregor Mendel is the father of genetics. And he was an Australian monk and he conducted his studies in the pea plant. Pea plant. Hope all of you know what is which one is this pea plant. And he conducted his studies in the inheritance pattern. Inheritance pattern. Inheritance means how one character is going from first generation to second and third or further generations. That inheritance pattern or the character in further generation, how they are studied. For that, he selected which plant? Pea plant. And he conducted all his experiments. So he explained the laws of inheritance. John Gregor Mendel explained, his father of genetics and Australian monk. He conducted his studies in the inheritance pattern. And he conducted all his studies in the pea plant. And finally, he explained 3 basic laws of genetics. Okay, 3 basic laws of inheritance or the pattern of that inheritance. So, the inheritance pattern and the 3 basic laws of that inheritance pattern, it was described by John Gregor Mendel, the father of genetics. Okay, and he conducted his studies in the pea. Then, see, next we are going to learn about. See, even though Mendel explained about inheritance and about the reason, he explained about inheritance, how the characters are getting inherited from one generation to next generation. Mendel explained about that and about the reason of that. Some factors, and Mendel called that reason as a factor. There is some factor that are going from one generation to next generation. Okay. So even though he explained all this inheritance concept, but the reason behind that, which he called as a factor, see, those were not very clear. Okay, that factor, what is that factor? That was not clear. That's why he mentioned the term factor, but what was the role of that factor? It is inherited from one generation to one next generation. That is clear. What is that factor, or which is that factor which is determining the inheritance? That was not clear. Okay. So in the next century, people concentrated more on this. What is that factor, or which factor is going from one generation to next generation, or how that factor is controlling all these traits when it is going from one generation to next generation. People concentrated or studies were more in the next century because they want to find out what is that factor means what. So the scientists gave more importance to the factors which is the genetic basis of inheritance. These factors, Mendel found out that these factors are the basis of that genetic inheritance, right? So what is that factor? That's what in the next century people were behind these factors. So later these scientists got an idea about the genetic material. Later they found out the genetic material, then structure of genetic material, everything. Then the structural basis of genotype and phenotype. All these they made an idea. In the next century, they got an idea about structural basis of genotype and phenotype. What is the basis of this structural basis? What is the structural basis of the genetics or genetical type of an organism, or the appearance of phenotype means the appearance, how it is appearing, that appearance of an organism. So what is the genetic fact behind this? And they got an idea about the structure, how the genetic material is, or what is the structure of the genetic material. So all these studies were going on in the next century. After Mendel's work, scientists were concentrated on all these aspects. So in the next century, they got lot of improved ideas about the term factor. So they made it clear that what is factor which is mentioned by Mendel. They got an idea about the factor that Mendel mentioned, okay, or of that factor or how it is passing from generation to generation. They got a clear idea. They learned through experiments. They conducted so many experiments and they came to a conclusion that Mendel mentioned factors, right? So these are not simply factors. They are the base of life. They got a conclusion that these factors which are mentioned by Mendel, they are not simply genetic factors. They are the basis of life, or they are the basic factors of the life. That means how it is becoming the basic factors that controls the inheritance and variation. So these factors are controlling the inheritance and variation. So the nature of those factors which determine the phenotype was not very clear during Mendel's time. Okay, the nature of that factors it was not clear. What is the nature of that factor which is mentioned by Mendel? It was not clear during Mendel's time. So these factors represent the genetic base of inheritance. In the next generation, the studies were more. In the next century, they conducted more studies and experimental evidences they made. These made a conclusion that these factors represent the genetic basis of inheritance. And understanding the structure of genetic material and the structural basis of genotype conversion became the focus of attention in biology for the next century. What do you mean by this sentence? So understanding, see, they got an idea about a factor, okay? So that factor later they concentrated on the genetic material which is having that experiments to prove that where these factors are present. So to find out that, they conducted studies for the genetic structural basis of the genetic material, then genotype, if a gene were genetic basis, and the phenotype of genetically the structure of the genetic material is one type, means how it is appearing or how it is giving the physical appearance, okay. Then the conversion from that genetic basis, the physical appearance, how what is controlling, and they gave more focus on these studies in the next Mendel's time. Then, so just now I mentioned about so many scientists. So the studies in the branch here in the genetics branch that reached molecular level also. They tried to find out the molecular structure of that genetic material. So all these studies were going on, or molecular level, up to molecular level they reached. So instead of superficial studies, they didn't make any superficial studies. Just like one character is passing from that, they want to find out the reason. So they gave a molecular level study, and there are so many scientists here. You can say number of scientists here. So there are so many scientists who made benchmark in this field. They gave important contributions in this field, in the field of genetics. So they are Watson and Crick, then Nirenberg, Corana, then Kornberg, Benzer, Brenner, and Monod and Jacob and Monod. Okay. So here Watson and Crick, Nirenberg, Corana, Kornberg, Benzer, Brenner, Jacob and Monod. So note down these scientists' name with their contributions. I'm going to explain one by one what and all they did for the field of biology. Okay. So first one is Watson and Crick. Here you can see the first picture, Watson and Crick. See what Watson and Crick did. Watson and Crick, they cracked the double helical structure of DNA. Double helical structure of DNA. You can learn about the double helical structure in the next chapter. So they conducted studies in this field and they presented the double helical structure of DNA. They presented like DNA is having, it is having a double helical structure with base pairs which this double helical structure of DNA, that two strands are anti-parallel to each other. All these informations they gave. So Watson and Crick cracked the double helical structure of DNA. And the details of double helical structure we learn in the next chapter. And next scientist is Nirenberg. Nirenberg was an American biochemist, and he was conducting his research studies in the field of genetics also. Next is Khorana, Hargobind Khorana. His full name is Hargobind Khorana. He is an Indian American biochemist. Okay, he is an Indian, basically he is an Indian and Indian American biochemist. Nirenberg and Khorana, they were conducting same kind of experiments. And in 1986, the year, note down the year, in 1986, Nirenberg and Khorana, they shared a Nobel Prize for breaking the genetic code. Okay, so first Watson and Crick, they cracked double helical structure of DNA. Okay, double helical structure of DNA, Watson and Crick. Note down. Next is Nirenberg and Khorana. Nirenberg and Khorana, they shared a Nobel Prize for breaking the genetic code. For genetic code. Okay. Next is Kornberg. Kornberg is an American biochemist and he won the Nobel Prize in Physiology of Medicine. Physiology of Medicine in 1959. Kornberg in 1959 he won Nobel Prize for discovering mechanism in the biological synthesis of DNA. Okay, mechanism of biological synthesis of DNA. In 1959, Kornberg won Nobel Prize for the discovery of mechanism in the biological synthesis of DNA. DNA biological synthesis. In that mechanism he studied how a DNA is getting synthesized biologically. So that mechanism was studied by Kornberg, and in 1959 he won Nobel Prize for this study. Okay. Next is Benzer. Next scientist is Benzer. He was an American physicist and molecular biologist. Okay. All are conducting their studies in the molecular level. Okay. They conducted their studies in the molecular level. Okay. And Benzer's contribution was in genes and behavior. Okay. He was conducting his studies in the genes. What are genes? Earlier, these genes were mentioned as factors. These genes only mentioned by Mendel as factors. John Grigor Mendel mentioned these genes as factors. So later they came to know that these are genes. Okay. So this Benzer, scientist Benzer, he was an American physicist and molecular biologist, and he made his contribution in the area of genes and behavior. Like single gene mutations in Drosophila. Single gene mutation in Drosophila. The Drosophila, the fly which is having, already it is having a genetic sequence, right? So if one single gene is changing, what and all the changes will come in the generation? That's what he was explaining. He was explaining about the single gene mutation in Drosophila. Okay, Benzer. Next is Brenner. Brenner, he shared a Nobel Prize in Medicine in 2002. In 2002, for which studies? For his contribution to this field of genetics. What studies he was conducting? Unraveling how genes control cell division. Okay. He conducted his studies in the cell division. Cell division. How gene is controlling cell. Okay. How division. Genetic level of cell division. Okay. How these genes are controlling the division of a cell. We know what is cell division. How these genes, each and every gene, how it is controlling the cell division, or what is the role of a gene in the cell division. That one is studied by Brenner. Okay, in 2002 he got a Nobel Prize for this. And the last one is Jacob and Moore. In 1965, not in the year 1965, they got a Nobel Prize for the discovery of regulatory genes. Okay, regulated genes or regulatory genes. This is the concept of operon concept. That concept is generally called as operon concept. That also you can learn in the next chapter. So in 1965, Jacob and Monod, they got a Nobel Prize for the discovery of regulatory genes. And the concept, that concept is called as operon concept. Operon concept. Okay. So in detail, all these concepts you can learn in the next chapter. So first, Watson and Crick, double helical structure of DNA, then Nirenberg and Khorana, Har Gobind Khorana. They shared a Nobel Prize for which work? Genetic code. Okay. And next is Kornberg. Kornberg, another American biochemist, and he got a Nobel Prize in 1959 for the mechanism in the biological synthesis of DNA. How the biological synthesis of DNA is taking place. He made studies in the biological synthesis of DNA. Then Benzer, next is Benzer, single gene mutations in Drosophila. Note down each scientist's name along with their discoveries. Okay, single gene mutations in Drosophila. That was Benzer's contribution. Then Brenner, gene, how a gene is controlling cell division. That was his studies. How the gene is controlling cell division. Then Jacob and Monod. That's for operon concept or the regulatory gene concept. Okay. Next is about James Watson and Francis Crick. Watson and Crick. See, that DNA model is called as Watson and Crick model of DNA. You'll get an model of DNA. You have get a question about Watson and Crick model to explain the structure, that and all in next chapter. Okay. So James Davy Watson, he was born in Chicago on 6th April 1928. Okay, just I'm giving a simple introduction because these are two important scientists in the field of genetics. So in 1947, he received BSc degree in zoology, then PhD in study of the effect of hard X-ray on bacteriophage multiplication. Okay, on bacteriophage multiplication, what is the role of how that X-ray, hard X-ray is affecting the multiplication of bacteriophage. On that field, he conducted his doctoral studies in 1950. Then Francis Harry Crompton Crick. Okay, Francis Crick. Full name is Francis Harry Crompton Crick. Okay, and he PhD in 1954. Again, almost the same concept, X-ray diffraction and polypeptide and protein. All were doing their research in the molecular level. Then these Watson and Crick friendship. And this Watson and Crick, both scientists met and they discussed about their common interest in the area of DNA structure and the molecular level studies. Then they made a serious effort, and the first attempt was a failure. It was not successful. And the second effort, which is based upon more experimental practices and experimental evidences, so that resulted in the proposal of complementary double helical structure of DNA. Okay, so what's that? They discussed about their common interest in the area of DNA and molecular level studies of DNA. Then they made an attempt, and the first attempt was successful. Okay, then they made another attempt, which is based on all experimental evidences. So that resulted in the proposal of the structure, double helical structure of DNA. Okay, double helical structure of DNA in 1953. Now down, double helical structure of DNA was proposed by Watson and Crick in 1953. Okay, it's important for NEET, no doubt. And here the honors to Watson with Crick. Okay, for this discovery, the honors, what and all honors they received, the John Collins Warren Prize of Massachusetts General Hospital in 1959. They got that award. Then Lasker Award in 1960. Then the Research Corporation Prize in 1962, and they got Nobel Prize in 1962 for this Watson and Crick model of DNA. That is the double helical structure of DNA. It is also called as Watson and Crick model of DNA. Okay. Next, let's learn about some genetical terms. So these terms which are going to use hereafter throughout the genetics portion. So in all these three chapters, in these terms, so you should know first the meaning of these terms. Okay, so all these are very important MCQs. You may get one or two definition of 1 mark question and MCQ, then 2 marks question to write this terms. So that kind of questions also you may get. Okay, important for board as well as NEET exam. So first term which we are going to use in this unit, or almost 3 chapters we are going to use these terms. First one is genetics. Genetics, you know, this is a subject that deals with the character. Okay, the characters which are inherited from parent to the baby, or from one generation to their next generation, or how difference is coming. We are going to learn about the characters first, then how characters are moving from one generation to next generation, or how changes are coming, or the same character, how many characters are going from parent to, or what is the percentage of this character which is moving from parent to offsprings. All these studies we are conducting under genetics, or that is a branch of biology which deals with all these inheritance and variation. Okay, the subject deals with the inheritance as well as the variation of characters from parent to offspring. Okay, genetics is the subject, deals with the inheritance and variation of characters from parents to offspring. So what and all we'll learn? Characters, how characters are going from one generation to next generation, or how difference is coming between parents and offsprings. All these we are going to discuss. We are going to discuss about inheritance and variation of characters from parent to offspring in genetics. Okay, so that is the subject which deals with the inheritance and variation. Next, inheritance. We'll learn about inheritance. So what is inheritance? The process by which characters are passed on from one parent to progeny. Okay, so the process by which characters are passed from parent to progeny, that is called as inheritance. Inheriting the characters are getting inherited from parents to offsprings. So this is the basis of heredity. So inheritance, in which pattern the characters are getting inherited. So all these are we are going to study under inheritance. We have to learn about the inheritance pattern of each character. So the inheritance or the heredity, it is otherwise called as heredity or the biological inheritance. How characters are inherited biologically. So it is a passing of traits from parents to offsprings, right? From parents, the character is passing to the offsprings. That may be through sexual reproduction or asexual reproduction. Whatever it may be, the character is passing from parent to offspring. And what do you mean by character is passing from one generation to next generation? The genetic information present in one organism, it is passing to the next generation, the same information or the percentage of that information. We don't know the level of percentage, but the characters, it may be some characters may pass 100%, some may be 50%. You can see that some tall plant and a short plant, if it is crossing means there will be some tall plants, there will be some short plants also, and in between medium height also will be there. So that is the percentage of that character which is passing from one generation to next generation. So the genetic informations are passing from parent to that child. Okay. Next is variation. Variation means, what do you mean by variation? Inheritance means the percentage of that character which is coming to the offspring. And what is variation? Variation means the degree by which the progeny differ from their parents. Okay, the degree by which the progeny is differ from their parents means it is actually the difference in the DNA. Okay, all characters are not passing from, I told no, just 100% of that character, it is not passing from one generation to next generation. So the degree or the percentage of difference between the parent and the progeny, that is called as variation. A progeny in if it is different, 50% difference it is showing from the parent, that is the variation. It is 50%, it is similar to the parental character. Okay, or it is similar to parent. And 50% difference it is showing. That difference or the percentage of difference from the parental organisms, that is called as variation. So it is actually the difference in the DNA among the individuals. So it is the difference in the DNA of the parental organism and the next generation, or the genetic information, variation in the genetic information. Okay, so there are so many sources for this genetic variation. That may be mutation, or some mutation means all these things you can learn in the second chapter. Mutation means there is some kind of changes. Okay, some kind of changes in the recombination. Recombination also you are going to learn a new concept in the next chapter. Recombination in different ways in the DNA, that is called as recombination. So all these things that also cause variation. All these are the reasons for variation. Okay. So what do you mean by genetics? The subject deals with the inheritance as well as the variation of characters from parents to offspring. All these studies. Okay. Next, inheritance means the process by which characters are passed from parent to progeny. Then variation is the degree of difference between the parents and the progeny. Okay. So next is factors. Factors already we told, Mendel mentioned these characters as factors. So unit of heredity, okay, which is responsible for the inheritance and appearance of characters. Now we are calling these factors as genes. These factors are the unit of heredity, or these are the unit of inheritance which is responsible for inheritance. Okay, the unit of heredity and characters passing from one generation to next generation. These are factors are the main unit of that inheritance. With these factors are responsible for the inheritance, and through the inheritance of these characters, or through the inheritance of these factors, the appearance of all these characters are possible. Okay, the factor, see hair color is controlled by a factor. So if that character is inherited as such, it will show same hair color, or the percentage, the difference in the percentage. Okay, that makes different kinds of hairs. So these factors were referred as genes. Earlier, John Gregor Mendel mentioned this genes as factors because he said some factors are controlling these things, these characters. So he mentioned that. Later, Johansson in 1909, he renamed, or he mentioned these factors as genes. Okay, in 1909, Johansson, he mentioned these factors as genes. Note down, genes. And Mendel used the term element or factor. Mendel used, which term he used? Element or factor to mention this. He used the word element or factor. He used that term. But later, this is renamed or referred as genes by Johansson in 1909. Then Morgan first used symbol to represent the factor. Morgan, the scientist, is a scientist, and he used the symbol. He used some symbols to represent these genes or factors. And dominant factors are represented by capital letter, and recessive factors by small letter. So who mentioned it like that? Morgan. Okay, Morgan used symbols to mention these characters or factors or elements. Dominant factors which are expressing in the next generation. Dominant in the sense, from parents, if you are producing one generation, in that generation some characters will be present, or the dominant, that appearing characters, that are called as dominant factors. they are represented by capital letters, and the recessive, the hiding characters, they are called as recessive factors. These recessive factors are represented by small letters. So who started this? Symbols and using symbols? Morgan. Okay, note down, Morgan represented like that. Next is allele. Allele means alternative form of a gene which are located in the same position or same. See, if DNA is there on same position, there will be alternative forms of a gene. A gene will be having one dominant form and one recessive form. Okay, a gene which is expressing one character, it will be having a dominant form which is dominating over the recessive character. The recessive character means a hiding character which is not expressing. Okay, if the dominant character is present, the recessive character cannot express on the homologous chromosome, that is called as allele. Okay, the alleles means different forms of same gene. Different forms of same gene. One will be dominant, one will be expressive, and one will be hiding. The hiding character is called as, or a hiding form of that gene, particular gene is called as recessive gene, and the expressing character or the expressing form of that gene, it is called as dominant gene. And here, see, Morgan used the symbol capital T, capital T. This is dominant form. Here capital T and small t. Here capital T, both are dominant forms. So here, if this is representing the character tall, that character it is representing. Here both are dominant genes. Dominant genes are expressed as capital letters. Dominant genes are symbolized as capital letter. We are giving a symbol of capital letter for dominant genes. Here, small letter recessive genes are mentioned as small letter. Okay, small letter. Here, then T is the dominant character. Capital T is the dominant character, and small t, that is also mentioning the same character, but that is the recessive. This is the short. This shows it is short. But here the dominating character or dominant character is present, the tall, which is mentioned as capital T. So the appearance or the phenotype or the appearance of that character will be this dominant character. That means it will be tall only. This character, the short one, that won't be expressing. Okay. Here both genes, both alleles in this gene, both alleles, one allele from this t, small t, and another allele the same at the same position, that another allele is also small. So here both are recessive. So it will form short only. And this term allele is coined by Bateson. Bateson coined the term allele, different forms of same gene, that is called as allele. Okay. So, is it clear? Next is homozygous. What do you mean by homozygous condition? Homozygous means if a zygote is formed by the fusion of two gametes. In sexual reproduction, you know, the zygote, which is formed by the fusion of a male and female gametes, that is having that identical characters or identical factors. It will be there. So these are called homozygote. Okay, and the organism which is developed from this homozygote, it is called as homozygous. Here you can see both. From the male gamete, you will get one allele, and from the female gamete, if you are getting another allele, both are in same condition. So that will express one character. Okay, same character it will be expressing. So these are in homozygous condition. Homozygous means the both are in similar condition. Here all these characters These are dominant characters, but in the last case, it is the recessive character, but that is also in homozygous condition. Okay, both alleles are expressing similar character, similar forms of a character. Okay, then heterozygous. Heterozygous, what do you mean by heterozygous? If a zygote is formed by the fusion of two different types of gametes. Here again the sexual male and female gametes are joining, but the character of that particular gene, it is different forms. That means these are different alleles of same gene. See, one is expressing the tall and one is short. Then definitely it will form a heterozygous. Then different alleles it will form. Okay, one is capital T and another one is small. But here the dominant character is present. So finally the phenotype or the appearance of that character will be the dominant character only. Okay, the recessive character will not express. And the individuals developed from that zygote, it is called as heterozygous. Okay. Allele is coined by Bateson, and the homozygous and heterozygous both are coined by Bateson. Okay. Here the term allele was coined by Bateson. Again, the homozygous and heterozygous term, these two terms, these are also coined by Bateson only. Note down this and all. The term coined by Bateson. Allele, homozygous and heterozygous. All these three terms are coined by Bateson. Next is hemizygous. Hemizygous, what do you mean by hemizygous? Hemizygous means if the individual that is containing only one gene of a pair, it is having one gene of a pair, then the individual is said to be hemizygous. Means male individual is always hemizygous for sex-linked genes. We have learned the sexual composition X, Y for male, right? While forming gamete X and Y. Then if it is a female, it will be XX, right? So the for male, it is in hemizygous condition. The sex determining gene in male. Okay. Next is phenotype. Phenotype means the morph of an organism. For a particular character, how the external appearance or the morphological appearance of an organism, that is called as phenotype. And the genotype is the genetic constitution for expressing a particular character, how the genetic constitution, the genetic makeup of an organism, how it will be and how it is expressing or the morphological appearance of that genetic constitution or the genetic makeup, that is called as phenotype, the external appearance. But at the molecular level or the genetic constitution of that particular character, that is called as genotype. Genotype. Next is another one is phenotype. Phenotype is the external appearance. Genotype is the genetic level appearance or genetic constitution or genetic makeup of a character. And these terms, genotype and phenotype, these are coined by Johansson. Note down, Johansson, he coined the terms phenotype and genotype. Okay. Next is hybrid vigor. This is another term which we are going to use in this hybrid vigor. So, what is hybrid vigor? It is the superiority of offspring over its parents, that is called as hybrid vigor. What is hybrid vigor? Superiority of offspring over its parental level or parents, that is called as hybrid vigor. And this develops due to heterozygosity. Okay, hybrid vigor down, it's developing due to heterozygosity. And This can be maintained for long time in vegetatively propagated crops. The hybrid vigor can be maintained vegetatively propagated crops. I like then hybrid vigor can be lost by inbreeding. Okay, we have learned this in the section of population. So that session we have learned hybrid vigor that can be lost by inbreeding or selfing, because inbreeding induces the homozygosity in offspring. Inbreeding or the selfing will increase the homozygosity. Always cross pollination is creating variations, right? Cross pollination or the selfing which is increasing the homozygosity. Always cross pollination increases variation, right? And the loss of hybrid vigor due to inbreeding, that is called as inbreeding. This term I have explained while teaching about that population explosion and all. So inbreeding depression. What do you mean by inbreeding depression? Loss of that hybrid vigor due to inbreeding. Okay, that is called as inbreeding depression. So actually heterosis, hybrid vigor, or it is called as heterosis. Heterosis or hybrid vigor. What is that? The improved or increased function of any biological quality. Improved function or increased function of any biological quality of a hybrid offspring. See, if two plants are crossing and hybrid is produced, that hybrid is having a better character compared to parents. If you are considering any one like or yield, that hybrid will be having more yield compared to both parents. That is called a hybrid vigor. So offspring is heterotic, or it is showing heterosis, that traits or the traits which are present, that is having a genetic contribution. Their genetic contribution, it is better the parental level. That is called as hybrid vigor. See, there is a slight higher production in the crop compared to the parents. That and all is both parents. One is the hybrid first generation from these parents. That is showing production compared to this. In the character of height, it is better than this parents, right? So that is a hybrid vigor. So that shows in the characters like size, growth, then then the animality, yield, in all these characters. So animal breeders or plant breeders, they used to exploit, or they are utilizing this kind of utilize by mating or by crossing different pure line. They will select pure line which is having a desirable or a better character, which is character which they will select character which is good in each parent, and they will do the crossing or the mating between pure lines. Then the offspring or the generation, next generation, it will show a better character or desirable character. Okay. So the first generation, great vigor. Okay, that's called, but this will decrease in the next generations, because first we are doing crossing. Next, the selfing is going on. So selfing will reduce that vigor will generations. So to maintain that, what we should do in each generation, we should cross for a new crop. If it is in the case of crop, for a new crop, we should cross in every generation to maintain that desired trait in that level. Okay. Next is about artificial selection. People were knowing about the differences or the inheritance and variation which is present in the sexual reproduction procedure, right? If sexual reproduction, that will create variations among offspring. So they exploited these characters, variations that naturally presenting variations which are inherited. After sexual reproduction, different types of offspring will form. So each and every one will be different in their character. So people started selection from these. The four are produced in that better character which is having better yield or better production and better vigor, they will select that one. So the desirable characters they have started to select. These are called as artificial selection. That is the artificial selection. We are selecting artificially. There is no natural selection. Are selecting. So domestication from the ancestral wild cows. Have such good Indian breeds which are artificially selected. See here you can see the selection. Flower in broccoli, the development is suppressed. Okay, so in intentional breeding, with that intention, some breeding process is going on. Okay, that is for the better character. In undomesticated banana, here this you can make out. It will be very clear from this example. The undomesticated banana, there was lot of seeds in that. Okay, there were so many seeds in that undomesticated banana or the wild variety, but the artificial selection through generation that made with no seeds. Very difficult to eat that banana, right? Everywhere seeds means now banana which is having no seeds, that one is selected artificially. And here for artificial selection, that Sahiwal cows in Punjab. In Punjab, this is special Punjab Sahiwal cows in Punjab. Note down Sahiwal cows in Punjab. This is an example for artificial selection. So now the Sahiwal, that breed is best dairy breeds in Pakistan also. In Pakistan also now these breeds are there. They are one of the best breeds in Pakistan. And Sahiwal, it is calm while milking, and that are the important characters. While milking, they are calm, and due to their they have been exploited to other Asian countries as well as Africa and the Caribbean places. Okay, so they have high milk production and heat tolerance. These are the characters of that Sahiwal breed. Okay, so these are the, Sahiwal breed is artificial selection. So that's all about the terms which you are going to use about genetics. So hereafter we'll be using these terms to explain. So go through these terms and read it well. Make the concept clear. So hereafter while explaining, I'll be using these terms. So it should be clear for you. If you have any doubt means you can ask. Is it clear? Hope up to this it's clear. If you have any doubt, I'll repeat. You want me to repeat anything? Deepa. Do you have any doubt? Okay, hope everything is clear. So we'll meet in the next classes. Is it clear? Do you have any doubt?
No, ma'am.
Okay. Okay, thank you. We'll meet in the next class.
Thank you, ma'am.